Plant hormones. The term hormones, when it comes into our side, but first comes the definition of hormones comes are the chemical messengers. Yes? Plant are the chemical messengers, signal transducing molecules that transfer a message or signals to activate a particular mechanism or particular pathways or particular signaling it does carry. So hormones are the signaling molecules or the chemical messengers. And next, or we got a definition. What it says is hormones are the organic compounds synthesized in one part of a plant and translocated to another part where in very low concentration, that is about 1 micromolar, it causes physical response. So this is a definition of a hormone. And when it comes to this definition, what three things we need to focus on, these three words that are given there. This is synthesized, that should be synthesized by the plant. There are different other products, there are different other compounds used by the plants that do promote or affect the growth and development of the plant, but they are not synthesized by the plant. But so what is the first characteristics or first necessity for something, the compound to be a plant hormone? It should be an organic compound and synthesized by the plant part. And the second one is it should be translocated. It should not necessarily, well, but it does. If so, the effect, what I meant to say is the plant hormone is synthesized in one part, and but its functionality is seen away from this part of synthesis. So it doesn't mean that it only shows effect away from the part it is synthesized. It does show effect on the part it is synthesized, but it has to be translocated to other parts as well. For example, you can take in case of ethylene when synthesized by the plants, it does cause the ripening of that plant fruit, as well as it spreads into the nearby fruits and cause the ripening of other as well. So that is what I mean. And after then, that it should be functional in very very low concentration, about 1 micromolar, and it should be causing a physical response. And in case of other compounds like the sucroses we talk about, they are synthesized by the plants, but they are not functional in the low concentration as the hormones are. So what I meant was it should be synthesized. What I mean by it should be synthesized means like calcium and potassium. These molecules are also utilized by the plants, but they are not hormones because they are not synthesized by the plant. And next one is that is what I meant. Sucrose is also but sucrose though is synthesized by the plants, but it is not a hormone because it does show the effect or growth only at relatively high concentration, that is about 1 to 50 millimolar, whereas the hormone can show the effect only in very less concentration, about 1 micromolar. So this is the basic introduction of what are hormones. So basically we're talking about plant hormones. So let's say what are plant hormones, what are the characteristics in order to be a plant hormones. Now well, let's see. Until now, we have learned about different plant hormones, and among them, the first 5, that is auxin, gibberellin, cytokinins, abscisic acid, and ethylene. They were the first 5 major classes of hormones that we'll be discussing about each of them briefly. And after then, that there's the 6th class also of hormone, that is the brachiosteroids, the steroid hormone that also does play an important role in the signaling pathways plus growth and development. And other two are this jasmonic acid and salicylic acids. These are the plant defense hormones. We say ethylene as well as jasmonic acid, salicylic acid. They are particularly the defense related hormones. The each of these hormones have their own characteristics and they have their own functionality. They have their own synthesis mechanism, and still they do have a interlink between all of this hormonal relationship. We'll be talking about it in the further lessons, but today we'll just be talking about how a simple hormone signaling transduction occurs. Okay, let's go. Well, see how these are the sites of hormonal activity, or let's say how a hormone is receptive. Well, okay, in this receptor, okay, in this site we'll see how a hormone is received by the plant parts, and then it is transduced for different signaling pathways. Okay, as you can see in here, wait a second. Okay, As you can see in here, this is suppose our hormone, any hormone it may be. The hormone is received by the receptors present in the plasma membrane. Most of the times we in this hormone signaling there should be a receptor, specific receptor for specific hormone present in the plasma membrane of the particular targeted cell. So when the hormones come in contact with the receptor of the receptor present in the plasma membrane, now what it does is, you can see in here, it activates the PLC, or we can say is phospholipase, that is present in the plasma membrane. So it causes the activation of this phospholipase C, PLC, phospholipase C, present in the plasma membrane. So when the phospholipase C gets activated, it helps in the hydrolysis, or it causes the hydrolysis of the membrane bound lipid, that is phosphatidyl inositol 4,5 bisphosphate, or PIP2, that is given in here. PIP2 is a membrane bound lipid consisting of inositol and glycerol group that is further hydrolyzed by the PLC, phospholipase C, on activation by the hormone bound to the receptor. And after the hydrolysis of PIP2, two compounds come in, that is, one is IP3 and the other is DAZ. IP3, also known as inositol 1,4,5 triphosphate IP3, and DAZ is a diacyl glycerol. IP3, let's see here, IP3 is a water soluble compound, so that is why the IP3 goes on and binds with the receptor present in the tonoplast of the vacuole. Why to the vacuole? Vacuole consists of large number of calcium ions within it. After the binding of IP3 to the receptor of the tonoplast, it causes the excretion of the calcium ions from vacuole into the cytosol. In case of animals, this phenomenon occurs in the endoplasmic reticulum, whereas in case of plants, the calcium ions are much more present inside the vacuole, and the receptor for IP3 are present in the tonoplast. So after the IP3 gets bound to the receptor, the calcium ions are excreted to the cytosol. Now as you can see, the calcium ion getting excreted out, the calcium ions get excreted, and it functions in activating the enzyme along with the that requires ATP for further metabolic activities. Like different enzymes may be involved in there, like ATP kinase or NAD kinase, or different enzymes get activated, and the metabolism activity is governed in that way. In other case, in the calcium ion is excreted in excessive quantity, four calcium ion binds with one calmodulin molecule, forming a calcium calmodulin complex, and this complex as a whole activates the multiple enzymes for carrying the multiple metabolic pathways. Now what we have all seen is IP3 from the hydrolysis of PIP2, the IP3 getting attached to the receptor. The remaining IP3 that doesn't get bound gets again further dephosphorylated, releasing IP2, then IP, and for again binds to the membrane, and the cycle goes on. The remaining DAZ that was remaining in here, IP3 being water soluble, went to the tonoplast, but DAZ is not water soluble, that is water insoluble, so that it keeps on binding to the membrane, plasma membrane. In plasma membrane, what it does is DAZ activates an enzyme in the plasma membrane, that is PKC, or protein kinase C. This enzyme uses the ATP to phosphorylate for the phosphorylation, causing activation and of the other enzymes. Like as you can see, it does the same functioning. PKC does the same functioning of is the same as calcium has been doing the functioning of the phosphorylase kinase activity. Protein kinase C, protein kinase gets activated, and it's simply activating other enzymes utilizing the ATP, and the further metabolic activities gets carried out. So this is a simple basic mechanism of a hormone signal transduction pathway. What you need is the receptor in the plasma membrane, the receptor in the vacuole, and then the enzymes associated with the lipid plasma membrane, PLC, PKC, and ATP. Obviously ATP is required for activation of different enzymes. So depending upon what sorts of site it is, depending upon what sort of functionality it is to carry, these enzymes are activated in that manner. So this is how simple way the hormones activate or conducts the signal transduction pathway.